Alright guys, we're still in chapter 1, and what we're going to look at here in this part of chapter 1 is going to be looking at the genetic disorders. And so before we actually get into the genetic disorders, we need to review a little bit about the principles of inheritance and how our genetics are passed from one person to the next. So the first thing here is Gregor Mendel was the first person who noticed the predictable transmission of traits from parent to offspring. He was an Austrian monk. He did work with pea plant. He was a gardener at the monastery, and so he started noticing this, and he started crossing plants to get different kinds of results. This led him to develop some terminology that we're going to look at. So one of the first things here is a phenotype. The phenotype is the outward appearance or the physical appearance of the individual or the organism. And so this is how we describe people, where we're like, well, she's tall with brown hair, it's curly, blue eyes, that kind of thing is what we would use for phenotype. Genotype is the unique genetic makeup of the person. And so the genes are what make her tall, or make her eyes blue, or make her hair brown and curly. But when we're looking at this, that's not how we describe her. We describe the phenotype, but the genes or the genotype is what determines that. These genes are found on the chromatids. Chromatids are two identical linear chromosome units. A lot of times when we're talking about them when we look at them inside cells, they form like an X. Okay, we make them like an X and separate during meiosis. They separate in order to create egg and sperm. The centromere is the center part of that X, and again, that is going to break apart in meiosis so that each chromosome can go to a side of the cell. We are considered diploid. This means that our chromosomes are paired. We get one set from our mom and one set from our dad, and they come together to form these pairs. One member of each pair comes from each parent. These pairs appear in what we call homologous or identical in structure, but they could have a different sequence on the DNA depending on what they code for. And so we would see on this piece of DNA from the mom and this piece of the DNA from the dad, they both code for eye color, but the mom passed on the blue code where the dad passed on the brown code. And so the code may look a little different, but they still code for the same phenotype. They still code for the same trait. All right, and so that's what we're looking at here when we talk about homologous chromosomes. Now, in us as humans, we have 23 pairs of chromosomes. Now remember, this is pairs, so this means 23 times 2, which gives you your total of 46. Out of these pairs, 22 of the pairs are considered autosomes. These autosomes are homologs, meaning that they have the same information on them. All right, and so on chromosome 1 from dad and chromosome 1 from mom will have the same information, but they may code for a different color of the trait or a different subtype of that, like straight hair versus curly hair. All right, but they're still going to code for that same kind of information. The remaining pair is known as your sex chromosomes. In females, they are still homologs because they have two X's, and the X's contain similar information. But in males, they have one X and one Y. The Y chromosome is totally different than the X. It has different traits on it, and this is what determines maleness. So when we're looking at this, guys, in females, they get an X from each parent, and in males, they get an X from their mom because that's all she can give, and they get the Y from their dad. Okay, so the males determine the sex of the child based on whether they provide the X or the Y. Now, when we're looking at the genotype, this results in 23 maternal and 23 paternal chromosomes. They unite during conception, so that fusion of egg and sperm come back together to create that diploid organism. All right, and you can see here that they've done some chromosome mapping. We've actually mapped the human genome, and so they know where all the different and where the genetic sequence for each of those traits is located. Now, if we look at the genotype, guys, and you get the same information from mom and dad, let's just say you got the exact same trait from them, brown eyes and brown eyes, you would be considered homozygous, meaning that you got the same trait from each parent. But let's say that you got brown eyes from dad and blue eyes from mom, you are considered heterozygous because they are different. All right, you had one brown and you had one blue. All right, a lot of times we represent the dominant trait with a capital letter, and we represent the recessive trait with a lowercase. This just means when it says it's recessive, that if the dominant is present, you will not see the recessive. The recessive will be kind of hidden, and it's still there, but it's being dominated by the other trait. All right, and so the example with the eye color is if you have brown eyes and blue eyes, the brown is going to dominate over the blue, and so they carry the blue-eyed trait, but you don't actually see it. Their eyes appear brown. All right, so one thing that we find useful, especially when we're trying to diagnose like certain kinds of genetic disorders, is using what we call a karyotype. A karyotype of an organism refers to a picture of its complete set of chromosomes. These chromosomes get arranged in their pairs, and they are going to be arranged based on their homologs, meaning that chromosome 1 from mom and chromosome 1 from dad go together, chromosome 2 from mom and chromosome 2 from dad go together, and so that allows us to see if there's any differences in those chromosomes. The only thing that you see that is different to determine sex is that if it is a male, like you see here, they have one X and they have one Y. But on the other hand, for the female, they have two X's. And so both of these karyotypes are, so what makes these karyotypes normal is that they have two of each of the chromosomes, because we should get one from mom and one from dad. However, on this slide, you can see that there are some differences where we see some things that are kind of haywire or wrong. So in this first set, they have 3 chromosome 21s. This is called a trisomy because they have 3 chromosomes. And this tells us that this has an extra chromosome that should not be here. This is actually how they diagnose Down syndrome. Okay, on this one, you can see that there's 2 X's and a Y. All right, when we look at this, guys, they have 3 of the sex chromosomes when they should only have 2. This is still a male because the Y is present, but having that extra X does potentially cause some problems. So this is also called a trisomy. Down here in the bottom one, this is called a monosomy. Monosomy means they have 1 of the chromosomes instead of 2. You'll notice that this individual has 1 X and that's it. They don't have another X or a Y. And so since the X is present, though, this individual is a female. All right, and we'll talk a little bit more about each of these in a minute. So when we're looking at genetic and developmental disorders, genetic disorders mean that the genetics have been altered or messed up in some way. Developmental disorders means that the development didn't take place like it should. That could have been due to the mom smoking or drinking or something happening that causes the development not to occur. So congenital disorders are present at birth. They could be genetic or developmental or environmental, or they could be a combination of both. Congenital malformations are associated with structural defects. This is due to the errors in fetal development. Frequently there could be a genetic cause to these, but sometimes it is environmental, and sometimes it's unknown of what actually caused that problem. Some inherited genetic disorders appear later in development, so they're not considered congenital. So you don't see them at birth, but because they are going to be present later, they still are considered genetic because they were in the genes, you just didn't notice them. Now these disorders get divided into groups. When we look at this, they could be apparent at birth or later in life. Majority are inherited from their parents. However, again, some can be spontaneous. They could just happen. And some can be from fetal development mutations, where something, a chemical or some sort of compound mutated the genetics. The genetics were right to begin with, but during development, something went wrong. Now, they can be divided into things like chromosomal abbreviations, meaning that they don't have the 2 of each chromosome like they should. We also see Mendelian single gene disorders, multifactorial polygenic disorders. Okay, so when we look at single gene, that means there's one mistake, but in a multifactorial or polygenic disorder, there's multiple mistakes. In a single gene but doesn't follow the Mendelian patterns, like with what Mendel found, this could be like a triplet repeat mutation, a mitochondrial mutation, or mutations that are affected by what we call imprinting. Now, these are all relatively newer things as we have learned more and more about genetics and we know more and more about DNA structure. We have found that our genetics don't always follow Mendelian genetics that he found with the pea plants. Now, in your textbook, there is a list of genetic disorders and their inheritance. If you look here, the single gene disorders can be divided into autosomal dominant, autosomal recessive, and X-linked recessive. So underneath the autosomal dominant, you have what we call adult polycystic kidney disease, familial hypercholesteremia, Huntington's disease, and Marfan syndrome. Now, we will talk about several of these in this chapter, but in some of these, we'll talk about them more specifically in the body system that they're contained in. In the autosomal recessive, you have albinism. Albinism is going to be the lack of pigment in the skin, color blindness, cystic fibrosis, phenylketonuria, sickle cell anemia, and Tay-Sachs disease. And then in the X-linked recessive disorders, you have what we call Duchenne's muscular dystrophy and hemophilia A. Now, guys, these all are due to one mistake in a letter, okay? One gene that is altered. So when we look at these are all disorders where there's one gene that's the culprit. In the multifactorial disorders, these are going to be things where there's multiple factors. They could be genetic and environmental or both. This would be like anencephaly, cleft lip and palate, club foot, congenital heart diseases, myelomeningocele, and schizophrenia. We also then have the chromosomal disorders, which are going to be where a whole chromosome or at least a large piece of a chromosome is missing or extra. These are going to be things like Klinefelter syndrome, Down syndrome, monosomy X, which is known as Turner syndrome, polysomy X, which is Klinefelter syndrome, and trisomy 18, which is Edwards syndrome. Now guys, one thing to note is this is not a complete list of chromosomal disorders or multifactorial disorders or even single gene disorders. This is just to give you an idea. So let's take a look at the chromosomal abnormalities first. And so these are generally due to an abnormal number of chromosomes, so they don't have the proper amount that they're supposed to have. So we're supposed to have 46 chromosomes as a human, and so they may have 45, they may have 47, they may actually have like 46 and a half because they have a piece of one that they shouldn't have. So when we look at this, it could be large pieces that are gone, or a whole chromosome is gained or gone altogether. This alters the structure of one or more of the chromosomes, and it's usually a result of separation during meiosis. So what happens a lot of times is due to what we call non-disjunction. This is when you see an aneuploidy where there's an unequal pairing. So non-disjunction is where the X we talked about is supposed to split and one goes into each side, and this is going to give you 23 in an egg, 23 in the sperm, so that when they come back together it makes 46. Well, what happens during the formation of the sperm and the egg is non-disjunction, where the X does not come apart, so the entire X goes to one side. This cell now has an extra copy, so instead of having 23 it now has 24, and this cell is missing a copy, and so now instead of having 23 it has 22. So if this cell was fertilized by a sperm, then we would have 22 plus 23, which gives us 45. If this one that had the extra was fertilized by the sperm, this would be 24 plus 23, which is 47. So it potentially causes us to have that mismatched number. All right, and so it can give us, like I said, between 45 and 47 chromosomes. If they're missing a chromosome, this is called a monosomy. This is where the daughter cell has a deficiency of one chromosome. This is usually not compatible with life. Most monosomies result in a miscarriage. Okay, the child does not have enough genes in order to form properly, and so because of this, a miscarriage occurs. There's actually only one monosomy that survives to birth. On the other hand, polysomies is going to be where there's extra chromosomes. They have too many. And again, this normally results in a trisomy, meaning they have more than one. So meaning they have 3 of one chromosome, 47 total. This may result in a viable fetus. It may go ahead and develop pretty much like normal where it survives. However, a lot of times they do have some severe disabilities due to this. Those involving extra or missing sex chromosomes may be debilitating. And so when we look at this, it's important to see which chromosome they have extra of, and this can determine a lot of times whether or not it's going to be a viable fetus. So the cause of this is really poorly understood. We're not exactly sure what is causing those chromosomes to not pull apart. However, we have associated some risk factors with this. One being advanced maternal age. Women are born with the number of eggs they're always going to have, and they're suspended in this state where they're getting ready to divide. And so the older they get, the more likely there could be a breakdown or problem with this division that takes place. And so maternal age is part of that. Also, if we look at this, abnormalities in parental genetic structure, let's just say one of the parents already has some issues with their genetic structure, it could potentially cause problems. And then if there's an abnormality in crossing over that takes place, when those X's come together like this, there is a chance where pieces of the DNA could be switched, and if they don't get switched correctly, then we could see problems. This is usually due to breakage and loss of or rearrangement of chromosomes, and it does happen during meiosis or even mitosis. In meiosis, there is that crossing over error that could occur where chromosome portions are lost, they're attached upside down, or they could be even attached to the wrong chromosome altogether. In mitosis, we see opportunities for chromosomal breakage and then rearrangement that can happen as well. And so if we look at some of the examples that were listed there, like Klinefelter is known as cry of the cat. And what they're missing there in Klinefelter is they're missing the long arm on chromosome 5. And so it's not missing an entire chromosome, but they're missing a big chunk of it, and so it's been lost. This causes the child to have some very distinctive characteristics. It gets its name because the child sounds like a crying cat. When they cry, it sounds like the cat is really in distress. It does also cause some learning disabilities and that, but it does result normally in a viable offspring. They do survive till birth. But we are going to focus a little bit on some of these where you get a whole chromosome difference, or if you're missing a chromosome. So the first one we're going to look at is a trisomy. This is trisomy 21. This is where they have an extra chromosome 21, and this is also known as Down syndrome. Now, Down syndrome is actually one of the most common chromosomal disorders, and it is one that we often test for during pregnancy. It is the leading cause of mental retardation, and there are some other characteristics that do come into play, like you can see here in the picture. Children tend to have a protruding tongue, low set ears, so their ears are a little lower. In the eyes, they can have what we call epicanthal folds. They tend to have poor muscle tone, a shorter stature, congenital heart defects or deformities, increased susceptibility to respiratory issues, and we do see that this increase of Down syndrome is related to advanced maternal age. Now, there are some other things that can be present as well. They can have what we call a single palmar crease, which is known as a simian crease. They can also have intestinal stenosis, where their intestines is not as wide as it should, which can cause digestive system issues. They also do have a higher predisposition to leukemia, which is a type of cancer. They have a larger gap between their first and second toes. And so there's structural difference as well as functional differences that we do see in their body due to the fact that they have an extra chromosome. Now, how do we diagnose Down syndrome? Well, we can actually do prenatal screening with this, where we can see if Down syndrome is potentially present before the baby's even born. What they can do is they could do an amniocentesis, where they can take some of the fluid out of the abdomen, and they can take some of the fluid out the uterus. This can then be tested for the baby's genetics, and we could do a karyotype to see if there's an extra chromosome 21. There are some less invasive ways now that they can test due to taking some of the blood from the mother, which then can help determine at least the chances. This would then let them know if they needed to go ahead and do an amniocentesis, because amniocentesis can obviously be a little more invasive. This deals with hormone levels and in their blood and stuff. And we also see they can do 4-dimensional ultrasounds now. There are some distinct characteristics dealing with the nose being more flattened and the back of the neck. They have more neck skin, and so that can actually be detected in the ultrasound. And so they can look at that and compare it to the blood test, and they can give you a pretty good idea if the baby has Down syndrome. Now, when we're looking at treatment for Down syndrome, there's no cure. We're not going to cure Down syndrome, but what we're looking at here is it's going to be symptomatic. So based on what symptoms they have, that's going to be the treatment they need. If they have holes in their heart due to their genetic deformities, they need to have that fixed. If they get a lot of respiratory infections, then we need to treat that. If they develop leukemia, then that's when we treat that. And so it's more of just treating when issues kind of come up or arise. And we also then need to also be supportive of the individual and their family, okay? Because these children, there are different ranges of the mental retardation, but a lot of times they're not going to necessarily be able to live completely on their own. And so the family as well as themselves need that support. Some other types of trisomies that aren't discussed here in this particular chapter are going to be things like Patau syndrome and Edwards syndrome. Both of these are also an extra set of chromosomes that can cause some major issues for the child. The next one we're going to look at is known as Turner syndrome. Turner syndrome is found in about 1 in 3,000 live births, but this is only going to affect females. So this is 1 in 3,000 live female births. The reason it only affects females is it's because they have one X. This is considered a monosomy. So they have one normal X and they are missing anything else. They have no Y and no other X. Therefore, this is a female phenotype. But because they only have one X, we do see that they have reproductive issues due to not having the two X's. The second X chromosome is missing or structurally abnormal, and this one usually comes from the father's chromosome. Okay, not always, but usually it does. These rarely survive to birth, but they could survive. This is the only monosomy that can. So some associated signs that come along with Turner syndrome are a shorter stature, so they're shorter in stature. They also tend to have a fold in the neck, which causes a webbed neck to take place. They have a wider chest, We also see they can also have congenital heart defects, and they have a failure of developing secondary sex characteristics. Now, this is because they have normally underdeveloped or missing ovaries. And since their ovaries are underdeveloped, it does not produce the estrogen and the hormones like they're supposed to. So this is going to affect breast development, fat distribution, and also potentially height. And so we see that they also can end up being infertile because they won't have menstruation due to the lack of hormones. We do see that they can have some characteristical facial features. The construction of their aorta could be abnormal. They could have an elbow deformity that can be seen on the outside. We also see that they can have smaller fingernails, a widely spaced nipples that are present, a lower hairline. And so these are all potentially some factors that come into play for Turner's syndrome. Now, diagnosis for Turner's syndrome, of course, is a history, a physical exam, and then they would do chromosomal testing. This is not one that they normally test for in an amniocentesis or in a blood test early on, but if the child appears to have these structures, then they would do a genetic screening on them, and then they could be diagnosed with Turner syndrome. Treatment, again, there's no cure for this, but we could offer them growth hormones as well as estrogen. This will help with the development of the secondary sex characteristics, but it may not allow for fertility. All right, and so because of this, the woman will normally still be infertile, but it can allow them to develop into some of the more normal female sex characteristics. The next one we want to look at is known as Klinefelter Syndrome. Klinefelter Syndrome is the most common sex chromosome abnormality that we have. It does affect males because this is going to be an XXY, and since the Y is present, it is affecting males. It's usually only one extra X, but sometimes it could be multiple extra X's. Abnormal sexual development and feminization is what kind of comes into play here. It causes them to be taller, have a slightly feminized physique. They may have a mildly impaired IQ. It's not going to cause them to be like mentally retarded, but it could cause them to maybe have a potential learning disability or something like that. Also, they're going to end up having like poor beard growth, normally lose their chest hair. They may develop other issues like breast development. This happens in about 30% of the cases. They could develop osteoporosis earlier due to their estrogen levels. They could also have more of the female type pubic hair pattern, and this is because of the extra X. They are producing extra estrogen and extra female hormones, and so because of that, it is causing them to kind of counteract the male hormones that are still being produced. Testes a lot of times will appear smaller, and this could cause some sexual dysfunction. Some other sex characteristics secondary sex characteristics that could be altered are their voice. Normally the extra testosterone is what deepens the voice of the male, so they may have a higher pitched voice, and they will potentially have hair growth distribution issues that are similar to more females than in males. When we look at this, again, diagnosis is going to be normally due to doing a chromosome screening after birth. It's going to be normally later in life. That's not going to be seen a lot of times when the child's born. It's going to be seen as the male starts to get ready to go through puberty and starts to develop a little bit differently than other males. The main treatment here is testosterone. Testosterone therapy is really important in order to make sure that this counteracts some of the feminization that could take place, but also the young man and their family may also need some support and some counseling. All right, so then this leads us to our Mendelian disorders, and we're going to look first at autosomal dominant disorders. Autosomal dominant disorders are going to normally be represented by a capital AA or a capital A, little a, okay, that you can see here. So it's dominant, meaning they have to have at least one capital letter and they have the disease. This is transmitted from an affected parent to the offspring regardless of gender. So if the parent is affected, there is a high chance that they're going to be passing this on to their offspring. This is a 50% transmission, which is very high. Unaffected do not pass it on, because if they are unaffected by the disorder, they're little a, which means they do not pass it on to their kids. A lot of times with these autosomal dominant disorders, we see a delayed onset that occurs. Some examples that we're going to talk about here in this chapter are Marfan Syndrome and Neurofibromatosis. There are other types that we will talk about or discuss later on in other chapters. So let's look at Marfan Syndrome first. So with Marfan syndrome, we see that this is actually a connective tissue disorder. There is a alteration in the collagen of the connective tissue, which causes some issues. This causes the individual to be typically tall and slender with very long, thin arms and legs. It causes them to also have very thin fingers, long fingers, and we call this arachnodactyly, because the fingers are very, very long and extended. We also see that they may have some eye issues. Retinal detachment and cataracts are a little more common, but one of the main things with the eyes is a lot of times they may just need glasses, which a lot of people who don't have Marfan syndrome also need glasses. The issue that becomes a major problem for the individual with Marfan syndrome is cardiovascular lesions. We see that the connective tissue is what gets weakened here, and with this connective tissue being a problem, we see that their aorta, which is the main vessel of their heart, tends to have weaker walls. This can cause floppy valves in the heart and aortic aneurysms, which can be life-threatening. So the aortic tends to be weak and it's susceptible to rupturing. If it ruptures, they can bleed out very quickly because it's the largest vessel leaving the heart. This is traced to a mutation in what we call the fibrillin 1 gene on chromosome 15. So we know that it's found on chromosome 15 and it is that particular gene. Now, there are some individuals in history that potentially may have had Marfan syndrome. One is Abraham Lincoln. If we look at pictures of him and things like that, exhibits a lot of the physical characteristics of Marfan syndrome. Another individual that's thought to have Marfan syndrome, it has not been confirmed, is like Michael Phelps. All right, and so when we look at this, it's not necessarily saying that it's a life-threatening thing that the kid is going to definitely die from Marfan's, but we do need to watch for those vascular issues that could be present. The next one is neurofibromatosis. With neurofibromatosis, these are neurogenic tumors. They're tumors that start to grow on the nervous system, on the nerves. This is a defect in chromosome either 17 or 22, and this is because there are two forms of this. Type 1 is a subcutaneous lesions that start to develop underneath the skin. A lot of times it's seen first with what we call cafe au latte spots. These are going to be spots that kind of look like coffee with milk, so they're like a light brown color on the skin. They will have normally at least 6 of these at birth, and they continue to get more and more as they grow. They may have excessive freckles, scoliosis, erosive bone defects, and nervous system tumors like you see here in this picture. On type 2, we see that the tumors are going to affect their hearing more, and it's going to have some acoustic nerve tumors that are going to affect their hearing. And that's the nerve that takes the information from your ear to your brain, helps you process. So it's going to cause hearing loss and even balance issues due to the inner ear. We also see individuals who have this issue could potentially deal with things like cataracts. They could also have balance problems. They could end up having ADD and speech issues depending on if there's issues in the brain based on if there's tumors, things like that. And so this can potentially cause some major problems, but a lot of times the issues that are seen more are going to be these lesions on the skin, these growths on the skin. The treatment is going to be more palliative, and it's going to be removing of the tumors. The reason we want to remove these tumors is that they can be precancerous, all right? It doesn't mean that they definitely are cancer, but it does allow for cancer to be more present if we're allowing uncontrolled growth. Also, it has to do with the aesthetic value and making the patient try to feel more comfortable. Again, we are not going to be able to cure this disease. All right, so now we're going to look at autosomal recessive disorders. Autosomal recessive disorders are when there's a mutation of the autosomal recessive gene. Males and females are going to be affected equally in this, and we usually do not see an issue in the parents. The parents are normally considered carriers of these genes, and so they're going to be that little letter and the little a, and so they could pass it on to their kid. 2 carriers have a 1 in 4 chance of having a child or an offspring that has the issue. If we see that they are both carriers, they also have a 50% chance of developing or having a child who's also a carrier. When we look at autosomal recessive disorders, we see that they are going to affect enzyme functions a lot of times, and so the enzymes become abnormal, which potentially causes problems for the individual. Some examples of these are albinism, phenylketonuria, which is also known as PKU, cystic fibrosis, and Tay-Sachs. So we're going to talk specifically about these 4. Again, there are other ones, but these are the 4 we're going to kind of focus in on here. So guys, with albinism is a lack of pigmentation. This is a disruption disruption of melanin synthesis. So the production of a protein melanin that's going to give you your pigment of your skin is hindered. And so they do not make a pigment in their skin. Certain kinds of albinism, which we'll talk more about in another chapter, can actually also affect the pigment in the eye and the hair. And so they have very light skin, very light hair, and they may even have almost reddish eyes. And the reason that they're red is it's a reflection, because there's no pigment in the iris. It's a reflection of their retina, which it appears red due to the blood vessels that are present. When we look at phenylketonuria, also known as PKU, this is an inborn error in metabolism. The child cannot metabolize what we call phenylalanine, which is an amino acid. The lack of this enzyme actually causes a buildup to take place. And so this is tested for at birth. When a child is born, they do a PKU test. They prick their heel, they take a sample of their blood, and they do the test. They will then do the test again when the child is about 2 weeks old. The reason is to make sure that once the child's been taking food in, whether it's breast milk or formula, that they're still able to digest this protein. And so they'll check for it again at normally 2 weeks. The child can appear normal at birth, but if this is not taken care of, it'll start to fail to meet certain milestones. Okay, it'll not be able to hold its head up when it's supposed to, or sit up when it's supposed to, and things like that. And it does cause progressive neurological damage. The buildup of this amino acid, because it's not being broken down, is toxic to the brain, and it will cause mental retardation if it's left untreated. The main way to treat this is the child is just put on a special diet. The special diet avoids this protein. And so it's very easy to treat if the child has PKU, and this is why we test for it, because it is definitely something that is treatable, that the child can live very long term if we treat it. The next one is cystic fibrosis. Cystic fibrosis is actually the most common autosomal recessive disorder. It's most common in individuals with Caucasian descent. We see that there's a production of abnormally thick secretions. So they produce abnormally thick mucus that's in their respiratory system and their digestive system specifically. With this, it's an actual defect in the tissue with the chloride transport across the membrane. And so it's one protein that has a defect that does not transport chlorine. And so because of that, it causes a buildup of that sticky mucus. And so they tend to be very susceptible to respiratory issues and illness. They also have digestive problems. And if left untreated, cystic fibrosis can result in death. Now, with treatment, we see that the life expectancy of a child with cystic fibrosis has increased greatly, but it's still only in the mid-late 30s. Okay, so the individual will only live normally mid-late 30s, depending on if they have complications that develop from the cystic fibrosis. The next one is Tay-Sachs. Tay-Sachs is a deficiency or absence of a certain enzyme that is called hexoamidase. This enzyme helps break down certain fats. So if you're missing this enzyme, these fats start to accumulate, and they accumulate in the brain. This destroys the myelination of the nerve. Okay, so nerves are myelinated with a special structure to help them conduct signals. This myelination gets destroyed by this fat being present, and so the nerves start kind of short-circuiting. Think about your wire for your cell phone or whatever. If there's a short in there, it does not charge the cell phone like it's supposed to. The electricity is going haywire. The same thing happens here in the brain. The signals start to go crazy. So the nerve cells actually start to deteriorate. They start to break down. The child will appear normal at birth. Then the infant will begin to miss milestones. It will progress into things like seizures, muscular rigidity, and blindness. And usually it's fatal by 5 years of age. There's no cure for Tay-Sachs as well. Now, Tay-Sachs is actually a big issue in the Jewish community. This has to do with the fact of the Holocaust. Anytime you lose large populations of individuals, it can increase the risk of certain kind of genetic disorders. And in the case of Jews, if they fully follow the Jewish culture, they're only going to marry other Jews. And so because of that, their gene pool is a little more shallow due to the fact of the Holocaust. Rabbis actually will do genetic screening in a sense, where they will look to see if Tay-Sachs is in that family, and they may advise against certain arranged marriages due to the fact that they may have a higher risk of producing children with Tay-Sachs. And Tay-Sachs is a fatal disease. The child will not normally live past the age of 5. All right, and so because of that, they may advise them to find another partner or potentially arrange a different type of marriage because they have a higher risk of having kids with Tay-Sachs. The next thing we want to talk about are sex-linked or X-linked disorders. These are going to be disorders that are associated with the X chromosome. The mutation of the sex chromosome, which is the X, is what we're focusing in on. Nearly all X-linked disorders are recessive, and females normally don't express this. It's very rare because they have to have a problem with both of their X's. Both X's have to be mutated. Whereas males, since they only have one X, you will see this as more of a problem in males. Affected fathers will transmit their gene to their daughters, and their daughters will become carriers. Their sons will be normal. Unaffected males do not carry the defective gene. Carrier females have a 1 in 2 chance of producing an affected son, and so when we look at this, guys, the males do inherit it from their mothers. Their mothers can also make carrier daughters. Again, females are rarely affected. So when we look at this, guys, one example of this is hemophilia A. Hemophilia is going to be a bleeding disorder. What they're lacking is factor VIII. This factor is very important in helping blood clots. When we see our blood clotting, it's like a domino effect. This is going to cause this, which causes this, and all the steps have to take place in order for a blood clot to form. What's happened here is you take one factor out, we start the blood clotting process, and once it hits that factor that's gone, it stops. It can't continue on with the sequence. All right, and so it causes a bleeding disorder to take place. And this is going to affect males more than females. This can be a life-threatening issue if treatment is not given. Another X-linked disorder is color blindness. You see males are going to be more often colorblind than females. However, color blindness is not a life-threatening problem. Another X-linked disorder we want to look at is called the Fragile X syndrome. This is associated with a single trinucleotide gene sequence that is found on the X chromosome. The individual will lack a protein that's necessary for neural tube development. Now, the neural tube is ultimately going to become the brain and the spinal cord. So what's happening here is, missing this protein causes it to not develop completely, and it does result in mental retardation. We also see some physical features that can be a part of this. The individual has a long face with a very large mandible, which is the lower jaw. They also have large ears, and since it does affect males, it's going to have larger testicles, mental retardation, learning disabilities, speech delays. They'll also have some connective tissue disorders and behavioral issues. So it's not just going to affect the sexual side in that sense. It's going to affect a lot of areas when we're looking at an X-linked disorder. The treatment for this, there's no cure. It's just supportive. Okay, it's supportive of the parents, but also the patient or the child. All right, now we're going to look briefly at the multifactorial inheritance disorders. These results from an interaction between the environment and genetic factors. During fetal development, the fetus and embryo are most vulnerable between the days of 15 and 60 after conception. Now, there's an issue with this. Most women do not even realize they're pregnant until normally around day 30. And then we're already in that period of the child being very vulnerable to potential issues. And so because of this, it's this early on part of the pregnancy during this kind of first trimester that they are the most vulnerable, because this time is when they're building almost all the different organs and structures. These are less predictable because one factor could affect one woman's pregnancy but not another's. We also see that they are extremely common, and they may be expressed at birth or even later. Some of the defects could be seen later. Some examples of these are going to be like a cleft lip or palate, like you see here in the picture, where it's a malformation of the top of the mouth. A club foot, where the foot is drawn in and pulled in. It's not straight like it should be. Congenital dislocation of the hips. This is where the hips, if you take the child and you put their knees up and you roll them out, their hips will dislocate. Congenital heart defects are part of this as well. Pyloric stenosis. The pyloric sphincter is a sphincter that connects the stomach to the intestines. Stenosis means that it's too small, so it does not allow food to come through. So you'd see the child not be able to gain weight very easily, and they may even have more reflux and spitting up because of this. Urinary tract malformations, where the kidneys don't form properly or the bladder does not form properly. A diabetes mellitus. So when we look at type 1 diabetes in kids, it is going to be a potentially multifactorial inheritance. Cancer in kids, and also psychiatric disorder. Now, when we're looking at psychiatric disorders, these might things like being more prone to depression, bipolar disorders, and schizophrenia, things like that. This is errors in fetal development that results in these malformations. The study of this is called Tetralogy is the study of developmental abnormalities, and a tetragen is a factor that causes these malformations. Okay, these tetragens could be things like chemicals, radiation, or even being exposed to certain viral infections. Those things may not affect the woman as she is going through the pregnancy, but they could affect the unborn child. Now, the susceptibility of the fetus or unborn child is going to depend upon the amount of exposure to those things. What developmental stage was it exposed during trimester 1, 2, 3, or all of them? Prior condition of the mother, if the mother had potential issues before, and the genetic predisposition of the fetus. The periods of most vulnerability are before the 3rd gestational week. Tetragen exposure either damages very few cells or so many that the embryo can't survive. And so when we look at before week 3, it could potentially cause a miscarriage relatively easily. Between the 3rd and 9th week, the embryo is very susceptible to these, especially on the 4th and 5th weeks, because this is when the organs are pretty much doing their development. After the 3rd month, the tetragens affect growth, or they may injure an already formed organ, and so they still could potentially be a problem. But we see here that the main time that they're most vulnerable is the 3rd to 9th week of development. Now, when we're looking at these terogenic agents, they will interfere either with cell proliferation, meaning making more cells, migration of the cells, or differentiation where the cells get particular jobs. Chemicals and drugs that can ultimately affect this are things like thalidomide. This would be a drug that they normally gave women who were dealing a lot with the morning sickness to try to help with that. They actually found that babies who were born with women who were on this extensively, especially from the beginning of the 1st trimester, we see that the child was born with pretty much no arms and legs. Now, when I say that, they had hands and feet, but they were just attached at the shoulder. The arm did not develop normally, just like the leg did not develop normally. Alcohol can also cause issues, and alcohol is a little different than some because alcohol can affect for the entire pregnancy. A lot of drugs will only affect during that 1st trimester. This is why when you go in, if you go into the doctor and you're told you're pregnant, they normally give you a list of medications that you can have and the list you cannot have. Those medications, guys, medications are put on a scale. They're looked at from A through D as well as X. If a medication is known as being A or B, it means that they're okay. There's a low risk or no risk of taking that medication while you're pregnant. However, when you get into C, it starts to potentially cause an issue, so we would want to probably stay away from it. We're not sure, but we want to stay away from it. But D and X are definitely problem drugs. You do not want to take drugs that are labeled as D and X if you're pregnant. And so because of that, we need to watch about what kind of prescriptions or medications, even over the counter, that are being taken by pregnant women, because they could potentially affect the development of the baby. We also see that there could be infectious agents like viruses, rubella. There's also a thing called torch, I'll talk about more in a minute, and toxoplasmosis. These are going to be things that could ultimately affect the baby. When we look at what we call torch, they're going to look at a number of, the T is going to be where you need to watch out for raw undercooked meat or like cat feces, because this is like the toxoplasmosis. We need to watch out for that kind of thing because it can affect the development of the baby and cause mental retardation and issues. And so being careful of what you eat and also cleaning out the cat box. The O deals mostly with a lot of the viruses like mumps, chickenpox, HIV, rash lesions. These could lead to blindness and deafness or failure to thrive for the fetus if you're exposed to those types of things. So when we look at the R, this deals with rubella. Rubella can cause hearing impairments, blindness, and even neurodevelopmental issues in the child. The C is cytomegalovirus. Cytomegalovirus can cause problems with brain damage, and it can cause retardation. And the H is the herpes infection. So herpes infections can affect the birth canal, and this can actually cause developmental problems in the child as well. So these are things that they would normally check for and see if you've been exposed to. If you're pregnant and you're around people with shingles, shingles is the same type of virus, and it potentially could affect the baby where it may not affect you. All right, and so that's why it's important to check those things. Also with radiation like x-rays, that's important because it can cause damage to the fetus. This is why when women go in to get an x-ray, they're normally asked, are you pregnant? All right, and it's not for your protection, it's for the protection of the baby. I want to spend a little bit more time on the fetal alcohol part because alcohol is one of those that can affect the baby through the whole pregnancy. From day one until the day before the baby's born, the alcohol could potentially affect the baby. It can cause physical, behavioral, and cognitive problems, and this is due to the mom consuming alcohol during pregnancy. The agent can cause harmful effects the entire pregnancy, and this is what makes it unique. Now, when we're looking at fetal alcohol syndrome, when we diagnose this, we're going to see that there are some particular things that they look for. They'll look for 3 facial abnormalities, and if you look here in the picture, there's multiple ones they could look at or use. These include things like microcephaly, the head being small, their low nasal bridge. They could have the folds like we saw with Down syndrome. Okay, so there's multiple things that we could see in the face, but they need to have at least 3 of these. We also see that they can have growth defects as well as central nervous system abnormalities. So these are things that are going to be looked for in that fetal alcohol syndrome. Now, the last thing is going to be with diagnosing and counseling with these multifactorial types of disorders. It's important to look at the fact that maternal age does cause a higher risk. So normally at the age of 35 or greater, they'll start checking for more of this stuff because it does cause a higher risk. Chromosomal disorders in previous pregnancies also put a higher risk for that pregnancy. A known family history of sex-linked disorders could potentially, again, put at a higher risk. If the family history, if we know there are inborn metabolic errors, neural tube abnormalities in previous pregnancies, this could be the anencephaly where the head does not form properly, or a neural tube defect where the spinal cord doesn't form properly, like in spina bifida. If you're a known carrier of recessive genetic disorder, you need to be aware of that in the sense of being able to pass it on. Ultrasounds can be used, and this is one of the non-invasive ways with diagnosis. A more invasive way would be the amniocentesis or the chorionic villus sampling. Both of these, though, guys, mean that we're going to actually access the amniotic fluid, which means it is a higher risk of potentially causing a miscarriage. The risk is actually relatively low nowadays with the amount of technology we have, but the risk is still there. We also see that we could do an embryoscopy. This is a direct visualization of the embryo, and that's where they actually put cameras in. It's not doing an ultrasound, they're putting cameras into the uterus in order to get a closer look at that child or baby. And so there are ways that we can look at this and potentially make a good diagnosis before the baby's even born, and maybe even fix some of the problems before the baby is born in some cases. And so these are just some of the things to kind of think of or look at when we're talking about potential genetic issues. All right, so this completes chapter 1. If you have any questions or concerns, please let me know.